Welcome to the new Chemist. We're glad you're listening. Feel free to download this podcast on Google Podcasts, Apple Podcasts, and Spotify. Here on the new Chemist, we discuss chemistry, which simply put is the science of change, as well as careers, community research, and COVID-19. We're happy you're tuning in. My guest today is Dr. Lewis N. Jungheim. Thanks for joining me today. It is good to hear from you. Just briefly, I'll inform my audience about you. Dr. Lewis N. Jungheim is a retired research fellow. He is the owner of L&J Consulting, in which its primary focus is the critical evaluation of potential in-license opportunities for big pharma clients. He is an affiliate associate professor of medicinal chemistry at Roosevelt University School of Pharmacy in Chicago, and he has served as a member of the Departmental Advisory Council and taught the medicinal chemistry of antibacterials. He is the former president of the Inuit Art Society. He was a medicinal chemistry consultant to the NTB Now initiative via Lilly's connection to the World Health Organization and the William and Melinda Gates Foundation. He is an accomplished inventor with 31 plus years of medicinal chemistry experience at Eli Lilly and Co. in infectious diseases, including antibacterials, antivirals, and antifungals. His medicinal chemistry experience includes oncology, neuropeptide receptors for neuroscience, and endocrinology. And he has also served as the inventor of the NK1 antagonist tradipitant. He was an NIH NCI postdoctoral fellow from 1979 to 1981 at the University of Rochester, New York, serving in Andrew Kennedy's research group. He earned his PhD in 1979 at the University of Wisconsin-Madison in Barry Trose's research group. He earned his BA in 1975 at Northwestern University at Evansville, Illinois. In 1992, he was elected the chair of the Gordon Research Conference on Heterocyclic Compounds, and he has served as an ad hoc member at the NIH study section in drug discovery and development. Highlights: while at Lilly, he served as the epigenetics platform chair, invented and developed small molecules that facilitate the oral uptake of therapeutic proteins. He was a member of the team that invented three clinical candidate growth hormone secretagogues for the treatment of frailty. He developed potent and selective inhibitors of MRP-1 transporters. He invented the first non-peptide-based inhibitors of rhinovirus protease. He was a member of the Lilly Argoron team that discovered Vericept, which is otherwise known as nelfinavir mesylate, for the treatment of HIV/AIDS. He invented novel cephalosporin-based prodrugs for the specific delivery of cytotoxic agents like vinca alkaloids and doxorubicin via tumor-specific conjugates. He invented the first potent broad-spectrum antibacterial non-beta-lactam inhibitors of penicillin-binding proteins. So these are just a few of his accomplishments. However, Lou is an accomplished medicinal chemist. Please welcome Lou Jungheim. Hey Lou, thanks for joining me today. It's good to have you here.
Good to see you again, David.
Yep. You have worked as a postdoctoral fellow, got your PhD from University of Wisconsin-Madison. You have served as a research fellow in many ways. What have been your longstanding interest in the field of science?
I guess, first of all, let's start with, I was always good at math.
Okay.
In school, and my dad was an electrical engineer.
Okay.
And so I think I always had a scientific bent. And while, let's say in Catholic school, science wasn't exactly the preeminent class, by the time I got to high school, I think I really loved my science classes the best.
Okay.
And had just a terrific chemistry teacher in high school too, which really kind of got me down that path.
Okay. So chemistry teacher. Okay, so would you say it was your chemistry teacher that kind of encouraged you and inspired you to pursue chemistry as an undergraduate and eventually graduate?
Yeah, absolutely. I think that was kind of my favorite science.
Okay, that's good. So, in terms of organic chemistry, what has been your focus? What was your focus primarily?
Well, my focus primarily was synthesis, making molecules.
Okay.
But as I got into my career, and I think part of the reason that I got into making molecules originally was because I think as an undergraduate, I really developed an interest in why do medicines work, and understanding that these were organic molecules that did things in your body that were of great benefit. And I think part of that was my junior year in college, I actually had an infection in my leg.
Are you serious?
And the doctor told me that if they didn't find an antibiotic that worked, they might have to cut off my leg. And so I think that was really kind of the beginning of my interest in why do organic molecules potentially do what they do in the human body.
Wow. Oh, that's an interesting story. So, so would you say, so your research was primarily focused on organic synthesis? That's good. So, given all of your accomplishments, how do you maintain view of the bigger picture in your career and in your life in general?
For me, that's actually been very easy, David.
Okay.
First of all, I think a lot of my colleagues always considered me to be potentially not a completely serious scientist.
Okay.
Part of that was I actually, as an undergraduate, financed my education as an athlete,
Mm-hmm.
and played all through college and progressed through playing semi-pro baseball in the Chicago area.
Mm-hmm.
In my youth, I've always had active interest in photography,
Mm-hmm.
creative things, and organic synthesis is certainly a creative thing. I love music, and travel and so on. So I've always had, and motor racing actually has been a fascinating thing. And part of that is because of my siblings.
Okay.
I've got a brother who's a mechanical engineer who actually works in the auto racing industry.
Okay.
So in fact, that kind of drove me to Indianapolis, where the speedway is, and the Indy 500 and so on. But it was also the best job offer that I got as well. So it was really a wonderful thing. Now, in terms of my career, that's also been very easy because drug discovery, hunting, medicinal chemistry is so multidisciplinary.
Okay.
That you really have to work with teams, people that are just as good at their own branch of science as you are at your ability to make molecules. And so I'm talking about experts in various fields of biology, pharmacology,
Mm-hmm.
pharmacokinetics, toxicology,
Mm-hmm.
and then even with physicians who are expert in the medical field for any particular disease that you're trying to target.
Okay.
And so you don't become an expert, but you have to become conversant in all of these different branches of science in order to really be an active participant in a team effort.
Wow. No,
But you have to be the guy, and that's the cool thing about the medicinal chemistry.
Mm.
The whole thing starts with an experimental molecule
Okay.
that the biologists start working with, and you start refining.
Okay. Yeah, that's good. That's good. Becoming conversant to be an active participant. That's good. So how have you been adaptive and creative in the field of science? What area, which niche would you say you have complemented to adding a layer of creativity or adapting a concept in a new way?
Well, that's easy because I've worked on so many different things.
Okay.
And I started out working on antibiotics.
Okay.
And maybe one of the most, well, I think I've done a number of creative things.
Okay.
The very first project that I worked on at Lilly, Lilly at the time was known as an antibiotics house.
Okay.
And kind of the game of penicillin, cephalosporin type antibiotics was drawing to a close, and there was the sense that really new things were needed.
Okay.
And actually in a collaboration with Edward C. Taylor, who was a professor at Princeton at the time, has subsequently passed away, but it's still a dear friend of mine.
Okay.
He had an idea where he was making square rings, 4-membered rings with 2 nitrogens and a carbonyl to mimic the beta-lactam, which only had 1 nitrogen. those turned out to be totally unworkable in terms of chemical stability.
Oh, okay.
And at the time, my first project was actually to try and make basically a backwards penicillin.
Okay.
So put the lactam in the 5-membered ring and really kind of squish it down with a 4-membered ring attached to it.
Hmm.
And Ted kind of inspired the idea of actually putting a 5-membered ring with 2 nitrogens in it.
Wow.
And those turned into a whole field of bicyclic pyrazolidinones
Wow.
that turned out to be very effective mimics of penicillins and cephalosporins and quite potent.
Wow.
And so these actually acted as beta-lactams, but weren't beta-lactams. I think maybe though my most interesting idea came later on when we were working on AIDS.
Okay.
And it turns out that this is where I first became exposed to a fellow named Anthony Fauci, who's been in the news of late,
Okay.
because he was kind of running and leading the way in fighting the world of HIV/AIDS.
Mm-hmm.
And I was part of a team that invented a compound called nalfinavir, which is an HIV protease inhibitor. And at one point in time was actually the top selling HIV protease inhibitor on the market.
Wow.
And the idea there was the molecule that kind of led the way came originally from Hoffmann La Roche.
Okay.
And there was some, and also this was at a really exciting time, because this was really when molecular modeling, X-ray crystallography, and quote unquote rational drug design was really coming into its fore.
Mm-hmm.
And there was the understanding that part of the molecule that Roche had a piece, a glutamine, in what they called the P2 pocket.
Okay.
And the mantra from Roche was that the only thing that worked in the P2 pocket was glutamine.
Okay.
And that was the kind of thing that sort of challenged me intellectually when people would say something that couldn't be done. So not unlike you couldn't replace the beta-lactam in a penicillin or a cephalosporin with something other than a beta-lactam, which I had already done.
Okay.
I started playing with this region of the molecule. And what I did was actually inverted the stereochemistry of the glutamine.
Okay.
And so instead of using an L-amino acid glutamine, I started making D-amino acids,
Okay.
where I put a moiety that could mimic the glutamine into the pocket where the glutamine was going,
Mm-hmm.
and the tail out into the rest.
Okay.
And so by having an unnatural amino acid, this really impacted the chemical stability of the molecules, because they were much less metabolically labile.
Okay.
And as we gained an understanding of taking natural amino acids out of a molecule, the glutamine was actually something that was really hindering oral uptake. And that kind of became the holy grail for the HIV compounds, and in particular the protease inhibitors.
Wow.
Was getting hydrogen bonding moieties and other such things out of the molecules. And it was working there and showing that these things could make very potent and better oral uptake molecules.
Wow.
That got the whole rest of the team working in that region. And ultimately those ideas led to an even greater simplification in the molecule that became nalfinavir, and ultimately a compound that impacted the lives of AIDS patients. I'm very proud of that.
Wow. That's good. That's good. That was very. That's good. I think you can dissect that some more, but we'll continue. That was good.
And these things are all published too. So
That's true. That's true. People can look them up. Yeah. So, how did you seek or find the right environment for you to thrive scientifically and intellectually? And what I mean by that is, you went to Northwestern, you went to University of Wisconsin-Madison, you are NIH NCI postdoctoral fellow at the University of Rochester, New York. You have spent a lot of time in medicinal chemistry. So obviously, in some way, shape, or form, you've had some success and you thrived scientifically and intellectually. How did you find the right environment for you to do that?
It's interesting. I did a lot of soul searching my third year, or at the end of my third year as an undergraduate.
Okay.
Because by then I had finished basically all the coursework required for the degree. And I kind of spent a good part of actually my junior year, thinking very hard about what I enjoyed most about chemistry.
Okay.
And given some of my other interests, like as a boy, I built a lot of plastic models,
Mhm.
and I enjoyed, working with my hands, other
Yeah.
such things. So it had to be a laboratory science. And then I look back and it was just organic seemed to be the best match for me, and also kind of the chemistry that fit. Also by then, and that was the time when I really had the infection in my leg that almost ended my baseball career.
Wow.
I ended up actually having my best season ever my junior year, right after recovering from that infection,
Wow.
and sought the guidance of actually Professor J.A. Marshall at the time, who was the best synthetic organic chemist at Northwestern at the time.
Wow.
And as an advisor, he agreed to take me on to do my undergraduate research project for my senior year.
Mhm.
But it was in talking to him and my saying that basically I kind of got involved in the thought process of, why do molecules work and so on. And that's where he said the beginning step there was really becoming the best synthetic chemist you can.
Mhm.
And that's what pointed me to synthetic groups.
Mhm.
And that led me to Wisconsin and working for Barry Trost, which I'm sure is a name that you know in synthetic organic chemistry.
Mhm.
From there, it became clear that the place to do the kinds of things that I wanted to do was in the pharmaceutical industry.
Mhm.
And that led me ultimately to Andy Kendy's group,
Okay.
because Andy had actually been a prominent medicinal chemist in the pharmaceutical industry before he left the industry to go into his academic career.
Okay.
And he offered me the opportunity to work on a molecule called Taxol.
Oh wow, you worked on Taxol.
In fact, I was the first person to put the tricyclic ring system together, and that was my NIH proposal.
Wow, you worked on Taxol. Wow.
Yeah.
Oh, that's cool, man.
And so it was the combination of Andy's recommendations, Barry's recommendation, and the relationship to Jim Marshall
Mhm.
that ultimately got me all of the job interviews that I really would have wanted to have. I got to interview for the big companies, and then being a Midwesterner, it was really great for me to be able to relocate to Indianapolis
Mhm.
and be near family in Chicago.
Mhm. That's good. That's good. Yeah, dude, that's good. So, given all your responsibilities and accomplishments, especially when you were busy, in terms of working as a postdoctoral fellow, working at Eli Lilly, how did you maintain a balanced life, or how did you try to maintain a balanced life?
Well, first of all, I had interests.
Okay.
All right. So motor racing, and boy, I moved to Indianapolis where
Okay.
there's a couple weeks in the month of May where you can really take advantage of that.
Mhm.
I'm an avid golfer.
Okay.
There are a lot of good golf courses around here. You
Mhm.
can do photography anywhere.
Okay.
But you come back to work, and I think part of the reason that I managed to be able to be productive, creative, and so on is because I had outlets for blowing off steam.
Okay. That's good.
I could take a day, and do something else.
Okay.
And so even though your mind is kind of always working, your head never shuts off.
Yeah, that's true.
In a lot of ways, you're always thinking about how to make this molecule and so on.
Mhm.
And I guess another point I would make is something that really contributed to my success was being a really good synthetic chemist.
Mhm.
There was no molecule that ever intimidated me in terms of trying to make it. And so I was always willing to try crazy ideas because we could usually find ways to make pretty exotic molecules to try wacky ideas.
Okay.
And understanding that in medicinal chemistry, if you've not made something that's never been done before, there's
Mhm.
no getting a patent, and therefore there's no getting a drug.
Okay.
And that's part of it too. But I think another side of it, and it's something that really got drummed into the heads of the people that worked for Barry Trost, was the importance of participating in the scientific community.
Okay.
And so it was always, when you can, publish. When you can go to meetings, go. And I think part of the interdisciplinary approach of medicinal chemistry and so on was, and of course you had the budget when you're working in industry to go to meetings that one kept you up with chemistry as a science, but two also helped you get much more acquainted and deeper into the various biological aspects of the particular kind of problem you were working on at the time. And to network with people that worked at other companies,
Right.
or other academicians who were working in the field. And that was really networking is just absolutely critical, not only within your company, but I think also the experts outside. And so I have lots of friends who worked at all kinds, at all the other big pharma companies.
Okay. Yeah, so that's good. So, in terms of your success, what would you say has been like the main contributor or guiding factor or guiding philosophy for your success as a graduate student, as a postdoctoral fellow, as a medicinal chemist in general? What would you say has contributed the most to your success?
My father used to preach to me as a kid, find something that you enjoy doing because you're going to be doing it for a long time. And so seeking guidance, seeking mentoring really mattered. But I would say the guiding principle through my whole career has been, and it's made me happy through the whole thing. You have a lot of opportunities to go in different directions in life as a chemist. The guiding principle for me is stick to what you're good at.
Yeah, that's true.
Even if there are other avenues, let's say like management, where you might make more money or maybe have more power, or things like that. It was for me, sticking to what I thought I did best is really what has kept me happy, productive, creative, and successful.
Okay. Wow, that's good. Sticking to what you know, sticking to your guns.
Yeah.
Yeah.
Because it's not like not like I ever wanted for money, excitement, fame, and wonderfully interesting things to do and great people to work with by sticking to being a scientist.
Yeah, that's true. Sticking to what you're good at.
Yeah.
So how did you maintain vision and teamwork in your environment? You worked as a postdoctoral fellow. I take it you worked in a team with a team of people, not by yourself.
And with many different teams on many different kinds of diseases. And that was one of the great things about it, because it got to the point where, because you were a good team player, other people sought you out to help with their projects. And so people were happy to have me join their efforts. And I think a big part of that was communicating with your coworkers, giving them latitude, letting them pursue their ideas as long as they continue to be successful. And I also think really respecting and listening to the experts when another member of the team is the expert at the moment for what they're talking about. And then supporting and standing by their feedback and their guidance, especially if you're the team leader. When there's not good news, you've got to stand up and say, as much as I wish that we were where, meeting the milestone or whatever, where we are now, we just can't because of these data. And a lot of that is just encourage the team to follow the data. It's a lot like what's going on in the world right now with the virus. Listen to the science.
Yeah, that's true. Follow the science.
Yeah.
So, in terms of advice, do you have any advice for those wanting to become medicinal chemists, wanting to serve in industry and eventually in academia? What advice would you give to people who are either in the undergraduate years or graduate years? What advice would you give to them?
Well, as an undergraduate, I would say do a lot of soul searching regarding what area of science interests you the most. Because there are so many different directions that you can go. Be careful how you choose where you go to graduate school.
What do you mean by that?
Be sure that where you go is going to give you the best possible exposure in the field you want to pursue.
Okay.
All right. There's a lot of things about, maybe, somebody wants to go to Harvard or Princeton or wherever, but that isn't necessarily where the best person or really good people in your field are practicing that science.
That's true.
Right?
Yeah,
and,
that's true.
and so you want to go somewhere where you will be able to get the best exposure and really good training in what you want to do.
That's true.
And then I think beyond that, the big thing is network. Take advantage of the people that are around you in graduate school. I mean, one of the great things about working at University of Wisconsin and in a group like Barry Trost was I've got friends all over the world now.
Yeah, that's true.
Because of people that I went to school with. And then network. Seek mentoring, and by participating in the scientific community, you end up meeting people in your field that work at other companies too. And those can be tremendous assets to you as you go through life. I mean, I was constantly getting calls by headhunters because of my contacts with other people.
Wow, that's good.
And so on. And so I always had plenty of other opportunities, but none of them ever looked better. I got really lucky and ended up in the perfect place for me right from the get-go.
Wow, that's good. That's good.
But I always had the chance to do other things, again, because of the networking that I had done and the friends that I had made externally. And that's both in academics and in industry. And it led to other opportunities. Like for 5 years, I actually consulted for the National Institutes of Health, participating in a study section because of the reputation that I had developed, actually both from people in academics and in industry.
Wow, that's good. That's good. Yeah. So, what has been some of the most beneficial advice you have received as an undergraduate, graduate, whichever area of your career you received the best advice?
I think I got terrific advice kind of every step of the way.
That's good.
As I said, Jim Marshall, who's unfortunately no longer with us, he really pointed me in the right direction of the way to understand best about what he heard me saying regarding human medicine, understanding molecules, and so on. The way to get into that was through the portal of synthetic organic chemistry, and becoming the best synthetic organic chemist that I could be. Because at the time, the best synthetic organic chemists that you can be were the people that Big Pharma were hiring to go into both either process chemistry or to drug invention. And for a long time, I thought, I might end up as a process chemist, learning how to most efficiently make new drug molecules. But I ended up, for whatever reason, and I think it may be because of the recommendations that I got from Trost and from Andy Kendi and so on, that the best fit for me was actually in drug invention. But again, with Trost, it was, of course, get your nose to the grindstone, be a participant in the scientific community when you have the opportunity, and network. And then with Andy, a lot of it was keep going, be creative, and publish every chance you get. And a lot of that can be even when you have a chance to, go to a meeting and give a poster.
Good. It's good. Yeah. So, Luman, thanks for joining me. I appreciate it. It was good to talk with you.
You too, David.
Thanks for listening. We're glad you were able to tune into this podcast. Once again, this is The New Chemist, where we discuss chemistry, which simply put is the science of change, as well as the other sciences, careers, community, research, and COVID-19. Thanks again for listening. Note, the views on this podcast represent those of my guests and I.